Hi, I'm Dr. Jennifer Strande. I'm a physician scientist. In particular, I'm a cardiologist and I research cardiovascular diseases. My main research focus is the heart disease of Duchenne and Becker muscular dystrophy. Muscular dystrophy is a genetic disease that affects mainly boys. So in muscular dystrophy, there's a mutation in the dystrophin gene, and when that happens, the protein, also called dystrophin, is not produced. So dystrophin is a large protein that serves as a shock absorber in the muscle cells, both in the skeletal muscle cells, the diaphragm, and the heart muscle cells. So when this protein is missing, the skeletal muscle, diaphragm, and heart grow weak over time. So we are interested in understanding the cause of why the heart weakens in muscular dystrophy and to find new treatments that might prevent or delay the onset of the heart injury in muscular dystrophy. As an undergraduate student at the University of Illinois in Champaign-Urbana, I took part in the summer research program. And during that summer I studied the neurogenesis, which is the birth of new neurons in honeybee brains. And my project entailed half the time I spent in the field observing the honeybees, and the other half of the time I spent in the lab studying the neurons, or the cells of the honeybee brain. So I always wanted to be a doctor, but after spending the summer in the research lab, I was hooked. And so going into a MD PhD program was a natural fit for me. When I was doing my cardiology fellowship, which is advanced clinical training in cardiology, I was asked to consult on a patient with muscular dystrophy. The patient was in the intensive care unit because he was having difficulty breathing or shortness of breath. And as I mentioned, muscular dystrophy affects the diaphragm, which is the main muscle that helps control breathing. But heart failure can also cause shortness of breath. And so the treatment team ordered an echocardiogram to look at how well the patient's heart was pumping. And he actually had a dilated cardiomyopathy, which basically means he had a large floppy heart that wasn't pumping as well as it should be. So wanting to treat my patient better, I searched the literature looking for treatments or therapies that I could offer him to make him feel better. And I was surprised to find that there were no specific therapies that have been shown to improve the heart failure that muscular dystrophy patients get. So as a clinician, I was limited at what I could provide the patient to actually improve his life. However, as a researcher, I have the opportunity to improve the lives of every patient with muscular dystrophy. We use induced pluripotent cells as a model system to study why the heart fails in muscular dystrophy. So to do this, we first collect urine samples from patients so we can isolate the urine cells out of these samples. So we grow up the urine cells, which we can reprogram them or modify them to turn into induced pluripotent cells. So pluripotent cells are master cells which can turn into any cell type of the body. So we're interested in turning these cells into heart muscle cells. It's a really cool process because these heart muscle cells beat just like the heart, except they're in the cell culture dish. I have a great research team working in the lab with me. This includes a technician, a postdoc fellow, and a graduate student. And while they have their own individual projects, they also help each other out on collaborative projects. I am really happy that they chose to work in my lab. It makes it very easy to come to work every day. I can work with great people, and we're all improving the lives of patients.